read the question, what the question is asking you, and then we'll be understanding the way of solving such questions. Okay, everyone read it properly now. This question go, but if you know the basic concept behind it, you'll be able to solve this question within maximum 2 minutes. Okay, let's see. See, the question is, you're given a certain number of cells that were taken in a tube, and a specific amount of 0.4% tyrene blue was added for staining. So we have a beaker, and in this beaker you have cells, and for staining these cells we have added typhine blue. Okay, about some percentage of the cell suspension was added between the hypocytometer and the cover glass for this particular, like this is the image given to you. The hemocyphometer is divided into 9 major squares of 1 mm into 1 mm size. The height of the chamber of the chamber formed with the cover glass is 0.1 mm. Empty circles indicate unstained cells, and the solid circles indicate the stained cells. Without having any knowledge about this particular question, we have understood just two things. First thing, that there's a beaker. In this beaker, we have some cells, and some staining content is added to it. Okay. Right, 100 microliters and 400 microliters. Now, about 20 microliters of this cell suspension was added. From this complete solution, you are taking some content and adding into the hemocytometer. So hemocytometer is what? It is a specific device which is helping in finding out the viability of the cells based upon what we have added, and to analyze the final result out of it. They have given you the result in the format of the circles. Like if you observe carefully this particular box, you're given two types of circle, some which are solid ones, some circles which are having empty circles, right, which is not having the stain in the middle. So the empty circles indicates unstained cell. That means this particular cell, if I take up this particular circle, is indicating my unstained cell. That is, this cell has not taken up the stain, right? But if I look into this solid circle means that this particular cell has taken up the stain. This is the information given to you. You need to find out two things. You need to find out that based upon this information given to you, you need to find out the total cell count in the original suspension. So if I have to find out, first thing is the total cell count, and the second thing that I have to find out is related to the percentage of the cell viability. Okay, these are the two things that I have to find out. So for both of these things, we have basic formulas to put in, very basic formulas to put in to get the entire calculation done. So before jumping into the calculation, did you understand the question? Is the question clear to everyone that what is the question asking you? Yes. Did you understand the question, everyone, till here? Yes. Now comes your work. What is the work that you have to do? We are not looking into this. We are just taking this particular graph and pasting it on the next, right? And enlarging it so that you can view it clearly. Now what you have to do for the first thing, like let's just remove this one, let's do the first part first. First question or the first pointer is you need to find out the total cell count. Total cell count, we'll be taking these four graphs, like this first one, this is your first one, this is your second one, this is your third one, and this is your fourth one. These are the four corners that you have to take into consideration, because this is actually showcasing your main result areas. Okay, so first, we'll be counting the total number of the cells in the four corners that are given to us. Okay, so everyone write down the total cells in this particular corner. So in the, let's say it is A, B, C, D. Okay, okay. So in the corner A, count the number of cells, irrespective of that whether they are the stained one or the unstained one. Count the number of cells in the first column or in the first square. This one. Everyone. Yeah. In the A column, count the number of cells. Very good. Everyone participate. 23 of you are there. All of you participate in the class. Yes. How many of them are there? One, two, three, four, five, six, seven, and eight. Right? So in my A square, we have eight of them. Count in the B. Count in the square B, everyone. Yes. How? Four, five, six, seven, eight, nine. Very good. Right? Now count the cell in the C. In this one, count the number of cells. Everyone. Very good. Five. And count in D as well. Very good. Excellent. So in D we have eight, right? So one, two, three, four, five, six, seven, and eight. So what is the total cell count over here? Total cell count is 8 plus 8, 16, and then you have 25, 30. So in total, we have 30 cells, right? Now, if I ask you that what is the average count per square, This is the 30 square, like 30 cells in the total complete cell count that we have done. What will be the average cell count per square? Yes. What will be the average cell count? How do we calculate the average? How do we calculate the average? Very good, Mamta. Anjali, how did you get 7 from this? And Preeti, how did you get 4.5? Not calculating properly, right? 30 divided by 4. Excellent. 30 divided by 4. What will be the answer? It will be 7.5. So this is what? This is your average cell count per square. Now, this is just your average cell count. What do we have to find out? We have to find out the total cell count that is given to you. So, how do we calculate the total cell count? So, for total cell count, you have a specific formula. What is the formula over here? Total cell count is equal to the average cell count in your square that is given multiplied by dilution factor multiplied by 10,000. Yeah. Why did we multiply it by 10,000? I'll explain it to you. Wait. So your average cell count over here is 7.5. How do we calculate the dilution factor? Before going ahead with calculating dilution factor, what do you mean by the term dilution, everyone? Yes. What do you mean by the word dilution? Think about it. Dissolving power. Okay. Dilution factor. I'm not dissolving something. Dilution. There's a difference between the term dilution and dissolution. That by what power are you diluting some content that is available to you? You were given 100 microliters of cells, and over here you added 400 microliters of a specific dye. That means total content was 500. You diluted it with 100. So that means what is your dilution factor? The dilution factor is 5 to reduce the concentration of solute. Okay, making liquid more weaker. Okay, we can call it that way. So dilution factor is 5 in this particular condition. Why? Because you have 100 microliters of the cell. In this you're adding 400 microliters of a dye, so total content is 500. So dilution factor will be 5 because you are adding 400 microliters, so 500 total. Adding 100 to it to dilute the entire thing. That's why the dilution factor will be 5. So I'll take the dilution factor 5 and I'll multiply it by 10,000. So 7.5 into 5 into 10,000. Why did I multiply it by 10,000? Because each of this specific cube has a volume of 0.1 cubic meters as given to you. So because every cube every cube is having the volume of 0.1 cubic meter, and 1000 cubic meter makes up 1 cubic meter, right? If I am multiplying it by like 1000 cubic meter is equal to 1 ml, right? You know this. So because you have 0.1 of them, that's why I need to multiply it by 1000. So it will be 10,000. That's why we have the average cell count over here to be calculated as average cell count multiplied by the dilution factor multiplied by the term 10,000. And when you solve it out, you will get the exact answer for the same. Anjali, I have not completed the thing, right? I have not completed the calculation. So wait for it. While I'm explaining, try to concentrate. So when you solve it out, you get the final value as 375 and then 3 zeros cells per ml. Okay, because you're given the formula, you're given the final answer in ml, and you're given the information in cubic meters. That's why we have to like make the calculation in the similar pattern. So for that, I have already multiplied by 10,000. And when you write it down in the format, and when you look into the four options given to you, out of the four options, you can eliminate third and fourth because you have reached to one of the format, one of the answer, that is the total cell count. Now coming to the calculation and coming to the entire explanation again, question is given to you as that you're given this information and you need to find out the total cell count. For that, first I calculated, I added the cell count given in all the four squares. When I got the calculation of all the four squares, I calculated the average. Average was because I had four squares. So total was 30. I divided by 4 and I got the average cell count as 7.5. Now comes the total cell count being equal to the formula is average cell count into the term dilution factor multiplied by 10,000. Why 10,000? I have explained. What is dilution factor? So dilution factor means that by what power, by what value do we have the dilution in the scenario, whatever we are taking into consideration. Over here you had 100 microliters of cells getting diluted. Like over here you are adding 400 microliters. So 400 microliters of the stain and 100 microliters of the cell. Total volume turns out to be 500. So this 500 total volume was diluted in a way with 100 microliters. That's why the dilution factor over here will be 5. So 7.5 into 5 into 10,000. When you calculate this, you get the final answer as 375 and then followed by 3 of the zeros. So this is the correct answer for, like, this is the first calculation of the question. Is it clear to all? Yes, Anjali, now is it clear to you? And others also, is it clear? 24 of you are there in the class right now. It's a very interesting question, and this is June 2023, like the latest paper question. So I hope the first part of the question is clear to all, right? Make sure that you, like, see, I have explained you it in a very elaborated manner. In the examination you will not require this much amount of time to solve it out, maximum a minute or two, that's it, right? So this was the first part. Now the second part of the question is to find out the percentage of the cell viability. Viability means the total cell, like the alive cells and the dead cells percentage we need to find out, right? So percentage of the cell viability is the total live cells divided by the total cells that you have multiplied by 100, because we have to calculate the percentage, right? Total cells we have are 30, total live cells. Now write down in the chat section, everyone, count the number of cells that are stained and count the number of cells that are unstained. Like from these 30, write down the stained cells number, everyone. Just see to it and write down the stained cell numbers. The empty circles is the unstained one, and the solid circles are the stained one. Write down the stained cells. Yes, 7 cells are stained. So the unstained cells are the one that are still alive, right? The unstained cells are still alive. So I have 23 divided by 100, 23 divided by 30 multiplied by 100. That's it. You get the final answer. Calculate, everyone, what will be the final percentage over here. Solve it out. Very good. It is 76.6, which is approximately 77 percentage. Right now, look into your options given. See over here, 375,000 cells per ml and 77 percentage, which we have just calculated. Okay, they have given approximate value, so we'll mark the answer in the approximation itself, right? That's why correct answer for this question is option number 2. Simple question, direct question. You just need to know what dilution factor is, what cell viability, and all these things are, right? I'm expecting the question is clear to all. Yes or no? Understood, everyone? Very good. Let's move forward and do one more question. Similar pattern. Okay. Now we'll try out this particular question. Okay. This question also looks very difficult on the first go, but it's very easy. Try it out, everyone. Think what is asked in the question, then I'll explain you what principle of parsimony is, and then we'll solve this question as well. Now the question. In the four taxas, two of the characters were scored to infer their phylogenetic relationships. The two character states were first was the shape, then was the color. Shape was either square or round, and then the color was either black or yellow. So, black and yellow. The character distribution is given to you in this manner. You're asked that the taxon is MNOP. Two of the characters are shape and color. You need to find out which of the following phylogenetic tree will be helping or will be uh, most suited as a or in the line of maximum parsimony. First thing first, what do you mean by parsimony? See, there's a concept in unit number 9, that is diversity of life forms, which is known as the principle of parsimony. It's a one line principle, but it's very, very important for you to understand. Okay. What does principle of parsimony says? Sometimes it is in the examination also, it is abbreviated as POP. So what is it all about? Principle of parsimony says that use the least number of steps possible. Yes, use the method that is simplest for you to showcase the characteristics between the taxons. That's it.